Metabolism is absolutely central to life. In order to fuel this process, humans take oxygen up out of the air that we breathe in and excrete the waste product, carbon dioxide. In Oxford, we have been developing a device to measure this process with unparalleled accuracy. There is a reason we've been doing this. There are opportunities to use these measurements in both healthcare and in the pharmaceutical industry.
I'm a clinician and I'm interested in patients with chronic airways disease. Now, one concern I have is that in contrast to other common chronic health problems, for example, ischemic heart disease, key outcomes have not improved much over the last 10 years in patients with asthma and COPD. We have a number of highly specific biological drugs that are becoming used to treat severe asthma and certain types of severe asthma. And I think it would be a huge benefit, not least for the economic and effective use of these drugs, to be able to identify clear evidence of a response relatively early on, before huge investment in terms of cost of drugs, but also patient commitment and clinician time. The second issue is for industry in the development of these drugs. They often have to do very large clinical trials with exacerbations as an endpoint. What they really need is an early readout that they can depend on and rely on, giving them a signal that the drug is having a biological effect early, and that being a catalyst for further investment.
We have used this device to make measurements of lung function in healthy individuals and patients with airways diseases. So these are healthy individuals. What you see here is the standard measure of lung function, FEV1, before and after salbutamol use. And as you see, there is no change seen. On the contrary, using our new measure of lung function, as shown here, at least in 3 out of 4 individuals, there's a significant reduction in the measurement. So this suggests that measuring lung function using this new device has the potential of being able to detect changes in response to drugs that we cannot currently detect using conventional lung function. So another thing we wanted to look at is whether using this new device and these new measurements of lung function, we are able to detect early changes in the lung, signifying early lung damage. What you see here is a group of healthy individuals who are non-smokers, and this is the new measure of lung function as measured by a device. And as you see, they are clustered together down here. Now, this is another group of people that are healthy but who smoke. And as you see, they are different. To put this in context, this is a group of COPD patients, and the measure of lung function using our new device clusters them up here. So what this suggests is that this new device is able to detect subtle early changes caused by smoking, which at the moment we're not able to detect using current lung function techniques.
We've seen we have a device that can detect disease in the lung early. We have a device that can measure really small changes in lung function with considerable sensitivity. This device presents a real opportunity for the pharmaceutical industry to conduct trials on smaller numbers of patients, making the development of drugs faster and cheaper. In terms of patient care, it gives us a way of looking objectively at what a drug does to their lung function. And this is going to be increasingly important to making sure we prescribe in a fair and effective manner, and so that we get the right drugs going into the right patients.